Sort of the broader message that I hope to achieve here today is that a comparative, a human comparative and a developmental perspective can really shed a lot of interesting insights on the dynamics of basic physiological systems that may matter to health, like inflammation. And I'm hopefully going to pick up on a couple of the themes that have been brought up by some of the speakers, in particular trying to reframe our understanding of life course and developmental processes, not as pathological when we talk about adverse early environments, not necessarily assuming that this is a model of pathology and damage like much of public health research does, but to think in terms of adaptation and developmental plasticity. And then the second theme that I'm going to pick up on, if I have time at the end of the talk about where I'm moving in my work to focus on a transgenerational model of health disparities and how inflammation may be a model for helping us understand how health in one generation can affect health in the next generation. Okay, so first, to get started with a little bit of background. Most of you probably know that inflammation is really bad for us, apparently, right now. Time magazine labeled it the secret killer. Everything that is going to do us in as we age has an inflammatory component: cardiovascular disease, diabetes, dementia, autoimmune diseases. But this is quite ironic and actually a relatively new conceptualization of inflammation, because prior to this recent attention on chronic low-grade inflammation, we understood inflammation as a key part of innate immune defenses, that if they don't function, you die. So inflammation has transitioned from a good thing to a bad thing. And so I'm going to try to reconcile those approaches a little bit in my talk today. This is the observation promoted by Paul Ridker and colleagues primarily at Harvard that has catalyzed this new attention to chronic inflammation and its contribution to chronic degenerative diseases. And the insight here comes from cardiovascular epidemiology, and what this graph is showing you that your level of high sensitivity C-reactive protein, a biomarker of inflammation, is an independent predictive risk factor for first heart attack in otherwise healthy adults. And it's a risk factor that's independent of your lipid profile, and that's just shown graphically here. Here's your lipid profile, and here's your quintile of high sensitivity CRP. A lot of research has replicated and validated this observation. And a lot of the early work to show us how we can measure this clinically and epidemiologically rests on what I'm calling the chronic low-grade inflammation model. This was a study that's highly cited and cited often by epidemiologists as justification for using a single CRP measure to predict future disease risk. And these are 4 people who were bled twice a week for a 7-week period. And you see there's a lot of between individual variation and not as much within individual variation. So there are 4 people. 2 people have consistently elevated concentrations of CRP. Oops, sorry. And then 2 people down here have lower levels. And this does not seem to be working. There we go. Okay. So there's separation here. And one sample gives you a sense of where people are relative to one another. And then physiologically, the idea is that adipose tissue in particular is a very important source of pro-inflammatory cytokines, but chronic infection, latent infections can upregulate these pathways as well. Pro-inflammatory mediators like IL-6 are produced. They go to the liver, stimulate the liver to produce and release CRP, which then has these downstream effects on pro-atherosclerotic processes, which are independent of lipids and blood pressure. Thank you. All right, so what do these two have in common? From the looks of him around the middle, he probably has pretty high concentrations of C-reactive protein, but so does the horseshoe crab, a species that's been on the planet since the days of the dinosaurs and does not have an elaborated system of specific lymphocyte-based adaptive immune defenses like vertebrates. Instead, it relies on pattern recognition molecules like C-reactive protein, which can bind to a wide range of highly conserved regions of pathogen. In fact, it was discovered in 1930 for its ability to react to the C polysaccharide region of Streptococcus pneumonia, hence the name C-reactive protein. So that's a different approach or perspective on inflammation and C-reactive protein that I'm calling here the acute responsivity model, where basically you have no CRP or very low levels of CRP in circulation. There is an acute stimulus, a pathogenic challenge, and concentrations increase dramatically by several orders of magnitude within 24 hours or so after contact with the pathogen. And then gradually concentrations come down over time as the infection is cleared. And so from this perspective, we have a pathogenic challenge, an inflammatory stressor which upregulates similar set of pro-inflammatory pathways, some of which cross the blood-brain barrier and induce symptoms we associate with infection. Liver goes to the liver, CRP production is increased and released into circulation, where CRP plays important roles in activating complement, promoting the activity of phagocytic cells, and opsonizing or tagging pathogens for clearance by other cells in the body. So very important roles in helping us fight infectious disease. So we have a model where we have actually dual domains of pro-inflammatory stimuli, body fat and pathogens, sharing a common set of cytokine-mediated inflammatory pathways. And we know that these are important for innate immune defenses against infection, and we think they may be important for chronic degenerative disease risk like cardiovascular disease. But all the research on this issue is done in people like this, what Jared Diamond called weird people, right? People in overfed, underinfected environments. And so we've really limited the range of human variation in research on chronic low-grade inflammation and undercut our ability to capture one of the key pathways that a priori we think should be relevant to the regulation of inflammation, being these pathogenic pathways. And for this reason, I've been doing research on inflammation over the past 6 or 7 years in the Philippines and in Ecuador. And the primary data set that I'm going to talk about today is in the Philippines, the Cebu Longitudinal Health and Nutrition Survey, which was begun in the early 80s, initially designed as a study of birth outcomes in maternal and child health and breastfeeding. There are high rates of infectious disease, acute respiratory infection, and infectious diarrhea in infancy at the time in the Philippines, and low rates of breastfeeding. So the study was designed to determine what are the factors that promote child survival and positive breastfeeding in an adverse lower income, lower middle income nation. The randomly selected set of neighborhoods were included in the study. Women were recruited into the study during the 3rd trimester of pregnancy. Their homes and their neighborhoods were surveyed during the 3rd trimester. The babies were followed up at delivery and birth weight was measured. And then every 2 months for the first 2 years of the baby's life, the mom and the baby were visited by an interviewer who collected information on morbidity, growth, breastfeeding, things like that. And then the study was designed to end. There were a couple of opportunistic follow-ups in childhood and in adolescence. And then I did a postdoc at UNC Chapel Hill where the study originated from. And it was about that time that the Barker hypothesis, DOHAD, early origins of adult disease stuff, started to come online. We realized that this was perhaps the best study in the world to look at the long-term effects of early environments on physiology and health, given the richness of prospective, objective data collection early in infancy. And in 2005, we mounted a major data collection effort, including blood collection, salivary collection, DNA, and other things. So I'll be talking to you about the results of that survey from 2005. In 2005, the participants in the study were 21, 22 years of age, and this is what their CRPs looked like compared to NHANES, widely recognized as sort of the gold standard in the United States for how the community is doing in terms of health. So I pulled an age and gender match sample of people from NHANES and compared their CRP to the Philippines, and overall, in the Philippines, young adults have CRP concentrations that are 0.2, 0.3 milligrams per liter compared to 0.9 or 1 milligram per liter in the United States. So dramatically lower. And this may be counterintuitive to you, since that I've just said that CRP is responsive to the infectious disease environment. The Philippines is a lower middle income nation with higher levels of infectious disease currently than in the United States. So perhaps they should have higher concentrations of CRP than in the United States. That's not the case. One possibility is body fat. Filipinos are leaner than Americans, even as young adults, and this is the mean waist circumference in the Philippines in our study and in the United States. Central body fat in particular is a very important source of pro-inflammatory cytokines, and that could perhaps explain population differences in CRP. But when we stratify based on waist circumference, we still see that Filipinos have CRP concentrations that are 50 to 30% lower than Americans with the same body mass, at least central body mass. So what's going on here? Now, a lot of people will ask me, this will be their first question, are there genetic differences? We know that genes are involved in the production of CRP, and the Filipinos have a distinct ancestry and population history. Perhaps there are higher or lower frequencies of genes that matter to inflammation. And, this could be the case. It reflects a more simplistic notion of gene to phenotype relations than most of us here would subscribe to. But nonetheless, I'm very pleased to have a colleague, a collaborator, Karen Mulkey at UNC Chapel Hill, who did a full SNP panel related to inflammation and other obesity-related genes in the Philippines and found that the frequencies do not differ substantially, and the contribution to explaining variation in CRP is quite low. So genes do matter in the Philippines, but they don't matter in a way that can explain away the major population differences in CRP that I'm trying to explain. So could this trace back to early environments? And could early environments have a lasting impact in shaping the regulation of inflammation in adulthood that's then reflected in the concentrations of CRP? So the model that I'm pursuing here is that in adulthood, most research, there's tons of research on cardiovascular epidemiology and emerging into social epidemiology on CRP in adulthood. And we know that body fat in adulthood, pathogenic exposures in adulthood, both increase concentrations of CRP. Could it be that there are analogous exposures in infancy, prenatally, or postnatally that may calibrate these systems in such a way that they have an impact later in life? So I'm looking for analogous things in the prenatal and postnatal environment. These are those things. I'm not going to go through them in detail for time, but these are things that we have measured in the data set, prenatally, postnatally at those 2-month intervals, and then we have analogous exposures in adulthood that we can also look at as potential pathways or confounders for the things of interest. All right, birth weight and CRP. So individuals born with larger birth weights in the Philippines have lower concentrations of CRP. And this has since been replicated in a couple of other cohort studies. And it suggests that well-established connection between birth weight and increased risk for cardiovascular disease later in life and across many studies could in fact be mediated in part by inflammation. So this is interesting, potentially important finding, but it cannot explain the population difference in CRP because Filipinos on average give birth to babies that are a couple hundred grams lower in birth weight than Americans. So if anything, they should have higher CRP based on birth weight and the prenatal nutritional environment alone. More interestingly, I found that 3 measures of exposure to infectious diseases and infectious microbes in the postnatal environment are associated with inflammation in adulthood. So this is the number of intervals in infancy that the mom reported that her baby had an episode of diarrhea, infectious diarrhea, over the week preceding the interview. So it's not a complete census of infectious diarrhea. It's sort of opportunistic. Every 2 months when the mom was visited, was that kid sick? And kids who had higher frequencies of infectious diarrhea in infancy have significantly lower concentrations of CRP as young adults. And here I'm modeling the probability of having elevated CRP in the top tertile of the population distribution. I can talk about why I did that. There are some particular statistical issues that led me to do that. So more infectious disease, more infectious diarrhea, lower CRP. This variable is based on the observation of interviewers who came into the participant's home, and this is a tropical environment. Most of the homes are open, have open walls in many cases, and domestic animals in some cases were allowed to roam freely into the homes, pigs, dogs, chickens. So the interviewer was observing whether animal feces was present on the floor of the home and whether the baby was at a stage where the baby could crawl. And so it's a proxy measure of likelihood of exposure to animal fecal material. So the more frequent that was the case, the higher level of fecal exposure in infancy, the lower probability of elevated CRP as a young adult. Philippines is also a highly seasonal environment with a dry season and wet season, and that is tightly associated with rainfall and transmission of infectious diseases. And a lot of the early work of the study showed the impact of breastfeeding on buffering kids from the effects of these seasonal effects on pathogen exposure. Kids who are born in the dry season have more infectious disease in their first year of life. So if you're born in the dry season, you spend more of that first year with exposure to infectious microbes than if you're born in the non-dry season. And 21 years later, you have a lower probability of elevated CRP. So we have 3 measures, each of which I wouldn't put too much stock in, but together they represent different types of information from different sources, all converging on the same conclusion that low levels of microbial exposure in infancy are associated with higher concentrations of CRP in young adulthood. And the results I'm showing you are fully adjusted for all of the variables in the model, including birth weight. So low birth weight also seems to be associated with higher CRP. And the mechanisms are not clear. This is a path forward for us in trying to figure out what specific cytokine pathways in particular or cellular pathways might be involved here. We're pursuing investigation of some epigenetic DNA methylation pathways as well. Will not be a surprise to anyone in this room. But conceptually, we can think about what's going on at sort of a higher level in different terms, and this is how I'm making sense of these results right now. In a high pathogen environment, you have, in infancy, many opportunities to engage with your pathogen environment. So that's what those red arrows are. Those are pathogenic challenges, microbial exposures. Your acute phase response, your inflammatory system, has the opportunity to upregulate and downregulate. And then to the extent that that practice is important, to the extent that there is some embedding of that in early environment, or to the extent to which the inflammatory immune system expects to engage with the environment to guide its development and function, then those experiences may carry forward into adulthood. And when you get exposed to an infectious exposure or other inflammatory stressor in adulthood, the system is very efficient at turning itself on and then turning itself off. In contrast, perhaps, in a low pathogen environment, these kinds of exposures in infancy during sensitive periods of development are less frequent. So the system has less opportunity to learn about its environment and to develop its regulatory systems appropriately. And then when an inflammatory stressor is engaged in adulthood, the response is more diffuse, it's overblown, or perhaps it's less efficient or less able to turn itself off once it's activated. So this is perhaps a conceptual explanation for what may be going on here. And so I'm trying to come up with a model, I'm calling it an ecological model, for how early environments might affect the regulation of inflammation. And so it's not just that early environments shape your CRP as an adult, they shape your immune phenotype, your inflammatory phenotype, and how you respond to inflammatory stressors to have sort of a very efficient, tightly regulated system to a more diffusely regulated system. So I've done 2 follow-up studies that I hope provide some support for this model. I'd be interested to see what you think. The first is that in those 2005 blood samples, we analyzed pro- and anti-inflammatory cytokines. IL-6 is tightly correlated with CRP. It's very important in promoting the production of CRP as well as other inflammatory processes. Did an analysis in the literature of all prior studies with healthy adults that have reported a concentration of IL-6. And that average from those studies is 1.91 picograms per ml. In the Philippines, our average is 1. So we have lower concentrations of IL-6. I mean, it's always challenging to compare across methods and labs, but this is consistent with the interpretation that there's perhaps some lower level of pro-inflammatory stimulus in the Philippines. But more interestingly and more importantly, I think IL-10, which is a very potent anti-inflammatory cytokine, actually at the cellular level prevents the production and activation of IL-6 and its impact on other cells. The average in other populations, although there are relatively few studies on this, was about 3. In the Philippines, we're at 7.5. So a much higher level of anti-inflammatory signaling, at least at the baseline condition based on our study. So there could be perhaps a different balance of pro to anti-inflammatory signaling here that may be relevant to understanding the CRP concentration difference that I showed you initially. All right, so this is the second study. I'm very excited about these results. These are just sent these off the other day, in fact. And we did a study in Ecuador to do a serial blood collection effort to look at CRP biovariability in the context of high levels of infectious disease. In environments like the U.S., where we know what CRP biovariability looks like. It looks like this, like I already showed you. We have no idea what CRP biovariability looks like in high infectious disease environments. And it could be that it's something like this. It could be it's like this. It could be it's like this, but everything's shifted up because there are higher levels of infectious microbes in the environment. We just don't know. So we sought to do this study and sent an intrepid graduate student to Ecuador last summer, and she collected 52 dry blood spot samples that we brought back to the lab for analysis of CRP. To orient you to the results that I'm going to show you, this is another way to represent CRP biovariability. And this is the same paper that I showed you before that's highly cited in epidemiology and clinical medicine as justification for using one CRP measure as an indicator of chronic low-grade inflammation. 26 people over 6 months, each dot represents their CRP at one point in time, and the line connects them. So what you see is you have clusters. You have individuals who cluster with relatively elevated levels of CRP. And this level here, 3 milligrams per liter, is commonly used as the high-risk cut point for incident cardiovascular disease. So there are individuals who cluster above, there are individuals who cluster below, and then there are individuals down at the low-risk level. And then you have some individuals with a lot of spread, so this is probably an acute infectious exposure, a flu or something. Normally this person would be down here. So there's, again, a relatively high degree of between individual variation and low levels of within individual variation. It's very important clinically in terms of measurements of CRP. So Lowland, Ecuador is where the study was conducted. It was conducted with the Shuar, an indigenous Amerindian group. They're horticulturalists, some hunting and gathering and scavenging, but they're relatively sedentary. Very high rates of infectious disease, unfortunately. 1 in 4 child deaths due to infectious diarrhea and acute respiratory infection, gastrointestinal illnesses, and vector-borne diseases are the primary sources of morbidity at all ages in this environment. So this is a pre-epidemiologic transition setting. We sampled 52 adults between 18 and 50 years at 4 weekly sampling intervals, and this is what we get. So this is individuals on the x-axis, their CRP on the y-axis, and I've sorted here by average CRP concentration by individual. And you can see, so this individual has 1, 2, 3, 4 values right there. And here's the 3 milligram per liter cutoff. Does this look different to you? There is no one in that category, right? So there are several individuals who produce a CRP value at one point in time above 3 milligram per liter, but every single one of those individuals also produces CRP below 3 milligrams per liter. In fact, all 3 of the other observations for that person in every case were below 3 milligrams per liter. So people can activate the system, but they can also shut it down. Look at the number of observations that are down near 0 here. It's a very different pattern of CRP biovariability. This is showing this again, but only for the 17 individuals who at one point during that 4-week study had 1 observation above 3 milligrams per liter, and you can see that things bounce around. So if you did a cross-sectional study at interval 2, 7 people would have high-risk CRP, but if you classified them as such, they would be misclassified because next week every single one of those people is below 3 milligrams per liter. So a very different pattern of production of CRP across time, which I'm interpreting as evidence for a different pattern of regulation, particularly with respect to the ability to shut inflammation down when it's not necessary. All right, so some implications and hypotheses in a couple of minutes here. So as I noted before, I think the way to think about early environments and their impact on inflammation later in life is not to just draw a straight line from early environments to CRP in an adult, but rather to think about the phenotype that develops and how the allostatic set points that may emerge from infancy may calibrate response to pro-inflammatory stimuli like these in adulthood, which then has implications for health. So it may not matter what your inflammatory phenotype is if these activating stimuli are not in play. If they are in play, then perhaps your inflammatory phenotype may matter, may be a moderator of the impact on health. So along those lines, can we think about the population differences in the relationship between body fat and CRP along these lines? Could it be that in Cebu, for individuals with the same level of body fat, there's a more robust set of anti-inflammatory pathways in place that keep inflammation in check, even though it does start to go up as you have more central body fat, but not as quickly as in the United States, perhaps because of these anti-inflammatory networks. We have the exact same pattern in our data in Ecuador. The sample size here is small, but again, you see an increase with body fat, but not the same level at each unit body fat of CRP as in the United States. A lot of interest in psychosocial factors and how they matter to inflammation, how stress affects inflammation, how depression affects inflammation, how inflammation may affect depression. A few years ago, I published a paper on older adults in the United States, in Chicago actually, where we showed that higher levels of perceived stress, Cohen Perceived Stress Scale, were positively associated with CRP. And so that was a nice finding. We have that instrument in our Cebu data set, and we find a mild positive association between perceived stress and CRP, so it's not statistically significant. But might the early environment moderate the association between stress and inflammation in adulthood? And I have some evidence here that that might be the case. This is very preliminary. This is on my to-do list for the upcoming year to write up. Individuals born in the dry season have higher levels of microbial exposure in infancy. Born not in the dry season, that's more like the United States, lower levels of microbial exposure. So at the highest levels of perceived stress, there is a significant difference here based on the quality of your early microbial environment. So some suggestive directions for future research. Most interestingly, I think for many of our interests here, pregnancy is actually a mild pro-inflammatory state. The dynamics of inflammation and immune function change during pregnancy for many reasons. We opportunistically had 80 pregnant women when we drew blood in 2005. I always removed them from my analyses because things were different. But I thought, this is an opportunity. This is a pro-inflammatory stimulus. Let's see what's going on. 24% of the women in this category, I'll get to this in a second, had low CRP. And these are just median splits because of small sample sizes. And then if I look at IL-10 as an important anti-inflammatory signal, about a third of the women had high CRP and low IL-10. I'm interpreting this as an out-of-control inflammatory state. Inflammation is up, but you don't really have the brakes on to turn it off quickly if you need to. This case, orange, CRP positive and high IL-10. Inflammation is up. Maybe it's doing some good things. It is adaptive. It's necessary for implantation. It's necessary for many things during gestation. But you'd have it under control because your IL-10 is up, perhaps. And it turns out that women who were born into homes with high versus low levels of microbial exposure have different profiles of inflammation during gestation. Women born in the high microbial exposure home in infancy, this is just a summary measure of the things I showed you before, are twice as likely to be in this well-controlled inflammatory state. Might this matter for birth outcomes? The best birth outcomes are for the women in that well-controlled inflammatory state. The worst outcomes are for women with high CRP and low IL-10, the poorly controlled inflammatory state. So this has provided preliminary data to support a grant application, which is going in a couple of months, using this intergenerational model of perpetuation of disparities in health with inflammation as the central mechanism. So there's a lot of interest in inflammation in adulthood and how it matters to risk for diseases of aging. If we really want to understand that inflammatory phenotype, we need to go back into the prenatal and postnatal environment of that generation, and perhaps we need to go back to the prenatal and postnatal environment of the preceding generation, because the inflammatory environment that a woman produces during her pregnancy is the earliest environment that the next generation experiences, along the lines of what Nancy Adler was talking about yesterday. So this may be a critical conduit for perpetuation of disparities in health across generations, and the things that we know matter, nutritional, infectious, psychosocial exposures, are highly patterned and partitioned by socioeconomic factors. So we're looking at those factors across generations in this grant application. Thank you very much.